The samples that we collect, when we take them back into the lab, we then start to cut them up and slice and dice them and make lots more samples, which we can then send to different collaborators for analyses, and we can analyze ourselves for different things.
Phytoplankton make this compound called DMSP, and then that DMSP gets released into the water. And there's tons and tons of bacteria, and they'll eat the DMSP and convert it into a gas called dimethyl sulfide, or DMS. And it's known as the anti-greenhouse gas. So this giant box is what I use to measure dimethyl sulfide. It's a lot more complicated and fancy than just a box. It's called a gas chromatograph, and it's really exciting for me to take it out to sea and have it be working out here in the middle of the Pacific Ocean. Is really cool. What I do is I will take these needles and then I just purge the sample with helium gas, and so it's bubbling the gas that's dissolved in my seawater sample out of the seawater and sending it onto this liquid nitrogen trap. And then I'll just take hot water, change the temperature very rapidly to send the gas onto the gas chromatograph. We are interested in measuring nitrous oxide, or N2O, which is a potent greenhouse gas produced by microbes in the ocean. About 1/3 of all nitrous oxide, or N2O, comes from the ocean. And we measure nitrous oxide using a GC, which is this machine here, and we collect our samples in glass vials and introduce the headspace, or a gas sample, into the GC, which then allows us to measure the amount of N2O in our water sample.
So I took seawater yesterday afternoon and I added nutrients to it, and I put them upstairs in the blue incubators and let them sit overnight and during the day today and try to grow some bacteria in them. So I brought them back here, and everything here is really clean to protect them from extra metals that can be added from the environment outside the ship, which is really dirty and rusty. Then we bring those samples back and we analyze the proteins that are in them, and we're going to try to see whether or not there's a change when we add certain nutrients, for example phosphate or cobalt, in the proteins that the organisms are making.
So we were looking for large plankton. We towed a 130 micron, 3-foot net. And basically what the net does is it channels all the plankton into the tail end of it, where we can then, collect them, concentrate them, and see what's there. I used the net to look for macroscopic prokaryotes, which in this particular station there were none. But what we got a lot of were eukaryotes. What you can see in this picture are 1, 2, 3, 4, one very large copepods that are still wiggling around and are quite alive. These are grazers. Basically, they'll eat anything that's smaller than them, and we have an awful lot of them in this particular net.
The time at sea is so precious, and we rarely get to get out here. So we have to make sure that whenever we're awake, that we're working, and whenever the weather allows us to get our instruments in the water, that we can do so, because otherwise we just can't collect enough material. But on the other side, it means that once we get back to our labs, back to Woods Hole, we're going to have a lot of work to do over the coming years, because we've got hundreds, maybe even thousands of samples.